and I'm here to present the person that will present our speaker. Because I would like to know I would like you to know who she is. So this is Chris Beninca, or Christian Beninca, and Chris is the director of the mitochondrial imaging core of the metabolism team. She's also doing research in mitochondrial genetic diseases, but she's a world expert in how to visualize mitochondria by all different types of microscopes. She has done an amazing job bringing this mitochondrial imaging core from one system to 4 different systems today that are all functional and ready for you to use. And she's providing the teaching of how to actually design an experiment to image mitochondria. So with that, I would like to invite Chris to present. Christian.
Thank you. Yeah, that's very funny. He always do that. But the reason is also because I know Christian from Cambridge. And I said, I want to invite him because I think his research is so amazing. And it was a great opportunity for us to just bring him all the way from Cologne, Germany here. So thank you, Christian, for coming, because it's a long way. So just an introduction about Christian. He's the group leader and professor of Metabolomics and Aging at the Faculty of Medicine in the University of Cologne in Germany. His research group is interested in understanding the contribution of dysregulated metabolism to aging associated disorders. His main focus is on cancer caused by mutations in Fumarate hydratase gene. Christian earned his PhD in biosciences in 2008 from the University of Padova in Italy, in which he focused on mitochondrial dynamics and apoptosis. Right after, he was the recipient of the prestigious Humble Fellowship when he moved to Beatson Institute of Cancer Research in Glasgow, UK. In there, he investigated the role of mitochondrial defects in tumorigenesis, and he became a group leader in 2012 in the MRC Cancer Unity in the University of Cambridge, UK, before moving in 2020 to Germany. Christian has made important scientific contributions in the metabolism field, and he has multiple publications in very important journals. You can see his CV is just populated by Natures and Cells. So it's a pleasure to have you. Thank you so much for accepting.
Thank you, Christian, and Orion for the invitation. I feel like it's a privilege to be here. Standing from there, I just realized there is a typo in my title. So this is fumarate hydratase, okay? So not fumarate hydrates. I don't know what that is. Okay, sorry for that. Okay, just give me 1 second. Okay, this is working. So as Christiane just said, our laboratory is interested in understanding the connection between dysregulated metabolism and cancer. But why are we interested in such a question? In the last 24 years, it has been demonstrated that mutations of core metabolic enzyme of the Krebs cycle, which I will name in a second, are the cause of hereditary and sporadic forms of cancer. These enzymes are fumarate hydratase, succinate dehydrogenase, and isocitrate dehydrogenase. In our laboratory, we are particularly interested in understanding the role of fumarate hydratase in cancer. And I just give a brief recap of fumarate hydratase. This schematic will appear many different times, so you will have the time to see it later. However, fumarate hydratase is the enzyme in the Krebs cycle that converts fumarate to malate. This is probably the more important information that you need now. Just for maybe those of you who are not familiar, well, this is a big mitochondria. The Krebs cycle is this metabolic node in the mitochondria that converts nutrient into redox equivalent that are needed for the mitochondria to produce ATP, amongst many other things. So in 2002, it was demonstrated that mutations in fumarate hydratase are the leading cause of hereditary leiomyomatosis and renal cell carcinoma. This cancer syndrome, which I call for brevity HLICC, goes as follows. Patients inherit one mutant copy of fumarate hydratase. So at the time when you see these patients, they are normal. However, during the course of their life, due to a process that is still poorly characterized called loss of heterozygosity, the patients lose the wild type allele, and this gives rise to a unique set of tumors, very tissue specific. So we have benign tumors of the skin and uterus. These are called leiomyomas or uterine and skin fibroids. These are benign, but they are extremely aggressive in nature because they grow and they need to be removed. And of course, this is probably one of the most important phenomena that occurs in this patient before they eventually they develop this type of renal cell carcinoma, which is called now fumarate hydratase deficient renal cell carcinoma. This is the most aggressive kidney cancer type that has been observed so far. Why I say this, that uterine and skin fibroids are probably more of a problem for patients, because whilst most of the patients affected by FH mutations develop skin lesions, just a proportion, let's say maybe 30%, will develop a kidney cancer. However, this cancer, as you will see later, this type of cancer is highly aggressive, meaning that when patients generally detect these tumors due to TC scan, it's probably already disseminated. And this is very different compared to what would happen in clear cell renal cell carcinoma. So now, okay, the transition is gone, okay. The thing I wanted to say here before moving into the more experimental part of the talk is that although this discovery of HLICC as the leading cause of fumarate hydratase, as the leading cause of HLICC was made in 2002, we still know very little about how and why fumarate hydratase mutation causes cancer. However, the first thing that I stumbled upon when I started my postdoc at the DGSOM with Eyal Gottlieb was something that probably relates more to the biochemistry and the role of the Krebs cycle and fumarate hydratase, meaning that we know that the Krebs cycle is a central metabolic node. And one thing that we couldn't understand at the time with Eyal, we were thinking, how is that possible that cells can survive without fumarate hydratase, and not only survive, but actually thrive and become tumors? So this led us to think, this was the initial hypothesis, that something must happen to the cells. There must be some sort of what we call metabolic adaptation that allow the cells to survive. And this is what I wanted to investigate, thanks to also at the time to the advent of metabolomics, which helped us to define possible metabolic changes that would compensate for the loss of fumarate hydratase. Just in brief, I want to explain a little bit the models that I'm going to use interchangeably. Don't be overwhelmed. This is just a schematic here. This is the first cellular model that I generated in a Eyal's lab. This is epithelial kidney cells from the condition on knockout mice. And important, keep this in mind, these are immortalized epithelial kidney cells. They grow. We generate knockout cells by infecting these cells with Cre recombinase. What matters is that we have two fumarate hydratase dependent clones, and then eventually over time, we also generate some fumarate hydratase rescue expressing fumarate hydratase in this fumarate hydratase knockout to assess specificity of what I'm going to show you. Now, more recently, Christina Schmidt, a PhD student at the time, developed also a human fumarate hydratase knockout cell line. These are, again, immortalized epithelial kidney cells, but we use CRISPR that now is widely available. And also here we generated two clones that are completely devoid of fumarate hydratase. The first thing that we observed, and this is probably now almost the ABC of cancer metabolism, but at the time, this was not a given. What we observe is that when you lose fumarate hydratase, one of the first and maybe obvious thing that would happen is that the mitochondria would be affected negatively by this. And in turn, this would give rise to what is now ascribed as aerobic glycolysis, or even in a term that I don't particularly encourage, is the Warburg metabolism, meaning that we have an increase in glucose oxidation to compensate for the mitochondrial dysfunction. This was, something in the textbook at the time. We used the sears to measure oxygen consumption rate, OCR, and extracellular acidification as surrogate for mitochondrial function and glycolytic activity. And as we would have expected, the fumarate hydratase knockout clones have very much decreased respiration and high glycolysis. So far, this is okay. Over time, we also demonstrated, we and others, that this increase in glycolysis is not only needed to generate ATP, but also for biosynthetic precursors, and this will be important in a second. The other very important phenomenon that we noticed at the time, again, this was the time where the first papers on glutaminolysis were coming, but the role was still unclear. But thanks to the work of Rafael Bernardini at the time and his help in this work, we were able to show that in the absence of carbons from glucose into the mitochondria, glutamine takes over as the major, we call it anaplerotic substrate. And glutamine fuels carbons of this truncated version of the Krebs cycle that starts with glutamine, ends with fumarate, and eventually we were able to show that this pathway is allowed thanks to what is called, we call at the time a carbon escape valve, whereby some of the carbons from glutamine are diverted out of the mitochondria and into this convoluted pathway called heme biosynthesis and degradation. And this eventually gives rise to this byproduct called bilirubin, which is the end product secreted in the cell, outer side of the cells. And we were able to measure this bilirubin production in the fumarate hydratase deficient cells. Keep this schematic in mind because it will come back in the next couple of slides. So what is important here is that we found this metabolic rerouting, and we proposed that considering this was happening exclusively in the fumarate hydratase knockout cell, because we could leverage this knowledge to selectively kill the fumarate hydratase deficient cells if we block this pathway. And indeed, this is what we found. This is our work many years ago. By knocking out hemoxygenase, which is one of the limiting step reaction of this pathway, we can selectively kill fumarate hydratase knockout cells. This is a colony survival assay when we knock out hemoxygenase. And this is what happens in the wild type. So this is an example. I want just to give it a, as a framework of thinking of how we can exploit metabolic adaptations specific in a genetic disorder here, or to kill selectively the cancer cell. And as you will see, this theme will appear also later. One aspect that we have been noticing whilst working on the metabolism of these cells is that this activation of glutaminolysis is really pervasive. These cells consume a huge amount of glutamine. And together with the production of ammonia that we know can be toxic, the cells use other reactions in order to spare the cells from ammonia production by glutaminolysis. And some of these reactions are indicated here. One of these reactions is asparagine synthetase. Again, keep this in mind because it will be important later. We have found that, again, this is ammonia production by fumarate hydratase deficient cells. You can see this is compared to the wild type, it's very, very much increased. But in order to allow this flow of glutamine carbons into the Krebs cycle, the cells even use reactions called transamidations, where some of the nitrogen of glutamine is given to, in this case, asparagine by asparagine synthetase. But another, and well, we can see here, this is just a heat map of a metabolomic analysis that we did recently. You can see that asparagine is very much increased in these cells. But also, importantly, we found that other reactions that leverage on this ammonia sparing set of pathway for the cells is the biosynthesis of nucleotides. This is very complex, and we haven't figured out yet how this all works. But this reaction is the first, at least, set of reactions. For instance, in pyrimidine biosynthesis, it's catalyzed by CAD, which is a tripartite enzyme. And we found that a lot of metabolic product of CAD are increased. Such as the first part of pyrimidine biosynthesis. However, and this will become clearer in a second, there is a problem with the later biosynthesis of nucleotides for other reasons that will become clear in the next slide. So what it means is that the needs and the craving from glutamate from the cells is actually not only allowed by the activation of glutaminase, but also leveraged by other ammonia-sparing reactions, such as nucleotide biosynthesis and asparagine biosynthesis. However, together with all this metabolic adaptation of central carbon metabolism, I just want to point out the key hallmark of the cells that will become, the hallmark biochemical and oncogenic of these cells is the accumulation of fumarate. Fumarate accumulates in both human and mouse cells, and this has now been demonstrated in many conditions to high millimolar levels in the cells. So fumarate really floods the cells. And of course, this has important consequences. From a biochemical point of view, would propose that this accumulation of fumarate needs to be buffered by cells. And what I mean by buffer, the cells need to take care of allowing fumarate to accumulate to a level that is sustainable for the cells. And to do so, they capitalize on the chemistry and the biochemistry of fumarate. And in brief, this is just a schematic, but I will explain a little more the details. Fumarate, for instance, is a metabolite that is not only produced in the Krebs cycle, but is also produced in a series of reactions in the cells, including the urea cycle, which is depicted here. And in a reaction called argininosuccinate lyase, the metabolite argininosuccinate in normal conditions in your urea cycle is converted into fumarate and arginine. What we found is that when fumarate accumulates to the high level, the equilibrium of this reversible reaction is shifted towards the substrate, argininosuccinate. And now the cells start to take up arginine to buffer fumarate, and they produce this argininosuccinate, which we found accumulated in the cells. This is another of these metabolic vulnerabilities that we could harness, because if we now, for instance, block arginine uptake, or we use arginine deaminase, that is called ADIPEC, which is a drug that is an FDA-approved drug to deplete arginine from the media, the cells do not have arginine to operate this buffering system, and they actually die selectively, as you can see this from this colony survival assay. Okay, another reaction that involves fumarate production is the purine nucleotide cycle. This is very similar set of reaction. The enzyme that produces fumarate from adenylosuccinate is called adenylosuccinate lyase, and is similar mechanism to what I show for argininosuccinate. When fumarate is accumulated, this reaction goes changes the balance, the equilibrium towards formation of adenylosuccinate. Now adenylosuccinate accumulates to very high levels, and this is another hallmark of the cells. What is important is actually why this is important. It was demonstrated very recently by Heather Christoph's lab. Why? Because this adenylosuccinate is not only, and adenylosuccinate lyase is not only present in the purine nucleotide cycle, but also is a critical enzyme in the purine biosynthesis. And what the lab showed is that when fumarate is accumulated, this biosynthesis of purine is impaired, and this leads another metabolic weakness of the cells. These cells become now dependent on exogenous supply, or de novo purine, or salvage purine metabolism. And indeed, they demonstrated by using 6-MP mercaptopurine, these cells can be selectively killed. And this, of course, is a very important, another important action for the patient. The other buffering system that I want to mention very briefly is now dependent not on the biochemistry of fumarate, but more on the chemistry of fumarate. And fumarate is a very interesting structure. It is an alpha-beta dicarboxylic, unsaturated dicarboxylic acid. What it means is that it is prone to nucleophilic attack from reactive cellular residues of cysteine, free cysteine, or from the cysteines in glutathione. And what happens is that this thiol group is now attacking this electrophilic carbon, creating a covalent thioether called succinylcysteine or succinylglutathione. And this, of course, is very important because now the cells use cysteine and glutathione to buffer fumarate. And this, of course, impinges on redox stress in the cells. Because now, if we constantly need the cysteine or cysteine uptake for creating more glutathione because this glutathione is used, then if we block cysteine uptake using, for instance, sulfasalazine, we could selectively kill, again, the fumarate hydratase knockout cells, but sparing the wild type. This is another example of how we could harness all this information, and this is all coming from mostly cell lines. I just want to point out that, of course, we are all aware of the problems with cell lines and how the work that we do on cell lines eventually mimic the disease. So here, this is a snapshot of how these findings will and have been validated also by other labs in patients and in mouse models. For instance, here, we found that in the mouse model where we knock out fumarate hydratase in the kidneys, the urine of this animal produced fumarate, succinylcysteine, arginine-succinate, and in the patients, in the tumor of these patients, we can actually see fumarate, and we can see accumulation of succinylglutathione, succinylcysteine. And in a very recent paper from Liang Zhen in Shanghai, they were able to show in the blood of the patients accumulation of succinylcysteine and succinyladenosine, which is a byproduct of SACAR in the purine metabolism. So this shows that actually all these metabolic changes that initially I said, okay, how do the cells survive, actually gave rise to important knowledge about how these metabolic changes can use as markers of disease, but also as metabolic vulnerabilities. Now, of course, this is what I started to be fascinated in the biology of HLICC, but there is much more to this that we need to understand. It is how does the loss of fumarate hydratase actually cause transformation? And we and others have focused extensively on the role of fumarate as we call a non-metabolite. Why? Because as you will see in a second, fumarate has the ability to elicit a variety of oncogenic cascades, and they depend again on the chemistry and the biochemistry of this metabolite. Don't need to show this again. Fumarate accumulates to very high levels, but one thing that I show you already is this reaction that is called succination, which initially I told you that occurs between free cysteine and fumarate, glutathione, and fumarate, but of course, this can occur also between fumarate and reactive thiol residues of proteins. This reaction is pervasive. Now, this is a volcanoplot of the succinated peptides, that are the peptides containing cysteine modified by fumarate in the human fumarate hydratase knockout cells. And you can see there's a significant number of enzymes and proteins that are modified by fumarate in the cells. This is quite exciting. I can imagine that you can see a lot of proteins that are very important for biology of the cells. We recently wrote a review about succination. Will be out soon, but this is the state of the art of all the proteins that have been found to be succinated in the fumarate hydratase deficient context. One that I want to mention more that has been investigated more in detail is actually that of KIAP1 and NRF2. So I'm here as a schematic whereby it has been demonstrated already more than 10 years ago by the lab of Patrick Pollard and Kyle Firth at the time, was that fumarate can bind to the reactive thiol residues of a protein called KIAP1. This is a very important negative regulator of transcription factor called NRF2. And NRF2 is a very important player in the antioxidant response. And this is where I think things can get very exciting. So fumarate can bind to KIAP1 in the cysteine residues. Then KIAP1 no longer keeps NRF2 out of the nucleus. NRF2 goes into the nucleus and it activates an antioxidant response that, amongst many targets, activate hemoxygenase, which you remember is one of the pathways that I told you is very important for these cells, but also glutathione biosynthesis. So here we have a very, I think it's a very cool example of how a metabolite can activate a transcriptional response that is needed to the cells to survive and to reach a new steady state. Now, the other set of reactions that I want to mention is that implicated in fumarate biology is the inhibition of a family of alpha-ketoglutarate-dependent dioxygenases. This is a completely different story. This doesn't depend on the chemistry of fumarate, but the ability of fumarate to block the activity of these alpha-ketoglutarate and oxygen-dependent enzymes. This is due to the fact that fumarate can block this enzyme by competitive inhibition. One of the most characterized alpha-ketoglutarate-dependent dioxygenases to date is the family of prolyl hydroxylases. These are enzymes that hydroxylate the hypoxia-inducible factor HIF, subjecting HIF to proteasomal degradation. What was found many years ago is that fumarate can block this reaction and lead to the aberrant stabilization of HIF. What it means is explained here. So fumarate can lead to the transcriptional activation of HIF and the target of HIF. This is just an example of how in our mouse cells, HIF is stabilized even under normal oxygen tension, which is something that doesn't happen very often. But what is important is the following, is that we were able to show that HIF can actually support the transcriptional reprogramming of glycolysis and angiogenesis. And for instance, one example, when we knock out HIF, this is still not published, but when we knock out HIF in the cells, the major consequences that we could find is actually the glycolytic arm. So HIF is very important for this part of the metabolic reprogramming, at least in part, by activation pyruvate dehydrogenase kinase, which eventually explains why we don't have too much carbons of glucose getting into the mitochondria. And the other very important aspect of fumarate hydratase biology is now linked to the inhibition of alpha-ketoglutarate-dependent dioxygenases involved in the epigenome. This is the case of TETs. These are enzymes that instead of hydroxylating proteins, they hydroxylate methylcytosine in DNA. And this has been very broadly speaking associated with changes in gene expression, activation of gene expression. So similar to what I showed you before, fumarate can block the DNA demethylation, leading to hypermethylation of DNA. And this, of course, you can imagine many important consequences for the cells. One thing that we demonstrated a few years ago now is that fumarate can block the demethylation of a family of antimetastatic microRNAs called microRNA 200. These are strongly suppressed in the cells, and this allow the repression of this transcription factor called ZEB1, which then eventually leads to a process called epithelial to mesenchymal transition. This epithelial to mesenchymal transition, which here is just represented by this activation of vimentin and suppression of the epithelial marker cadherin, can explain, for instance, the aggressive nature of these cells, because EMT has been associated with, and the mesenchymal feature of the cells being associated with metastasis, which is a key feature of this tumor. So now here we have a very intriguing scenario where when we lose fumarate hydratase, we have a prerequisite is to undergo metabolic changes that are required for the survival. And then when fumarate accumulates, eventually it activates a series of oncogenic cascades. One thing that we were pretty much unsatisfied from this picture, beside the fact that it's very complex, is that it doesn't tell us much about the relationship between all this reaction. What I mean by this is that when we take a tumor that has been there for a decade, or a cell line that has been adapted in condition in the media upon fumarate hydratase loss, all these signatures appear at the same time. And we don't know which one is actually a pro-survival, which one is pro-tumorigenesis, and which one is pro-metastasis. So we wanted to take a different approach and almost imagining putting another dimension to this, which is the time. So we started to take this into what we call, the chronology of fumarate hydratase loss. We want to understand what happens over time when we lose fumarate hydratase to see at what point, or actually how these reactions that I mentioned are orchestrated. But this is even more important now because if we now take another dimension, that is the tissue where this occurs, we could probably have even a better understanding of the tissue specificity of HLA-CC. What I mean by this is that all the work that you have seen was performed in kidney cells. And therefore, by and large, we don't know, all these metabolic adaptations, all these responses can be very specific to kidney cells. And we don't know what happens in the other tissues and actually how the other tissues react. And this is what we have been doing lately. So just give a snapshot again, we approached this question from two different points of view. First, we use pharmacological inhibition of fumarate hydratase, which is now available. We can use a fumarate hydratase inhibitor, and therefore we can see what are the very, very acute consequences of fumarate hydratase loss. And I put this because it is intriguing for us. We found that just after 24 hours, there is a comprehensive transcription and reprogramming. But the first thing that appeared is actually what is now called the mitochondrial integrated stress response. And we could see that this is dependent on ATF4. And what is intriguing is actually ATF4 is a transcription factor that activates the asparagine synthetase that you remember at the beginning was a way for the cells to allow uninterrupted glutamine flow, but also a variety of enzymes in purine and pyrimidine biosynthesis. So we think that we think that ATF4 actually is a very important mediator of the nitrogen metabolism in the cells. But going more into the tissue specificity response, and then just, I think we're doing okay with time, right? Yeah, okay. So what we wanted to perform now is look more broadly into the tissue specificity question. And to do that, we needed to generate a new mouse model. And this is just a representation. We used, as you remember, the flox allele for fumarate hydratase, and we crossed it with an inducible ROSA 26 Cre-ERT2. What it means in a nutshell, without going into details, is that we can knock out now fumarate hydratase by giving tamoxifen injection in multiple tissues in the mouse, adult mouse, and we could actually track the events in multiple tissues. So I'm going to show you now what we found by comparing a variety of tissue, looking at very early time points. Now, instead of 24 hours with the mouse, we couldn't just go from day 5 to day 10, and the results started to be also quite exciting. So what we found is the following. This is just H&E staining, here's a methoxylin staining, to look at how simply the tissue responds to tamoxifen and to fumarate hydratase loss in a different way. And just bear in mind that here I'm showing the tissue. We have confirmed that the loss of fumarate hydratase is complete in all this tissue to the same extent. But look, the kidneys, this is where fumarate hydratase loss causes cancer. They are not affected by fumarate hydratase loss. The tissue architecture is preserved. But look at what happens instead in the small intestine is a dramatic reorganization of the villi. Actually, from someone who is an expert in the small intestine would say that these villi are completely disrupted. So there is a very different effect if we compare the small intestine and the kidneys. Now we took the colon, for instance, just to make sure that this was not something that happened to gastrointestinal tract. We found that actually the colon is not as affected as the small intestine. So it seems that fumarate hydratase loss is very severely affecting the small intestine, but has minor effect in the kidneys. And this, of course, has important implication that I will summarize in the next slides. Just wanted to show you that even the metabolic response is very different between the tissues. For instance, if we look at fumarate and 2-succinylcysteine, which is our readout for fumarate accumulation, we found surprisingly that in the kidneys, fumarate is not accumulated until, or not even strongly, at day 10. But what accumulates is the fumarate buffer to succinylcysteine. But to look at the small intestine now, in the small intestine, the accumulation of fumarate is very significant. And as it is the 2,6-system. So it seems now that there is a different metabolic hardware playing in the small intestine and in the kidneys. And what this means in a nutshell, and this is the interpretation that we have, is that there are tissues that are permissive, such as the kidneys, where fumarate hydratase is allowed because of some potentially metabolic changes that are allowing this adaptation. But in other tissues, such as the small intestine, fumarate hydratase loss is not tolerated. And if we have, for instance, in this case, in a patient, one cell that loses fumarate hydratase, probably this cell would die, and this tissue is considered non-permissive, and this is why we don't have small intestine tumors in these patients. This is just a speculation on which we are currently working on. Besides this differential response that we see in these tissues, we started to look at the transcriptional response to see whether there were actually convergent phenomena. And this is where we got surprised again. This is a volcano plot of the gene signatures that occur in the fumarate hydratase knockout kidneys, small intestine, kidneys, small intestine, and colon. And we found something that we did not expect at the time, is that together with some of the other cascades, such as hypoxia and antioxidant response, the more enriched signature was actually inflammation. And when we plot all the genes that were belonging to these inflammatory signatures that we never found, actually we found this EV202B, which was accumulated already at day 5 in all the tissues. And this is where we got intrigued. I'm not going into details about this, just to give you some information about why we were intrigued by EV202B, because EV202B is a cytosolic DNA scaffold that actually is implicated in inflammation via at least 2 pathways, the AIM2 and the CTSI, which I'm sure you've heard about, which gives rise to this inflammatory response via interferon response genes. So to cut a long story short, we wanted to understand why there was an inflammatory signature in the cells, and we found we generated a reductionist approach. We took the cells from this condition on knockout animals. We knocked fumarate hydratase out by giving tamoxifen. And this is probably interesting to the mitochondriologists here. When we look at how the network of the mitochondria change over time, we started to get intriguing, maybe to explain why the cytosolic DNA sensitive signatures started to be important. So look at the mitochondria. This is a magnification of the mitochondrial reticulum when the cells were absolutely fine. When we lose, we give tamoxifen, something happens. First of all, the mitochondria start to become very swollen and large. But then when we look at DNA to see whether there was any cytosolic DNA sensing cytosolic DNA being released by cells either from the nucleus or the mitochondria, we started to see that in the lot of the very swollen mitochondria, there were what we speculated at the time and then eventually demonstrated, DNA released from the mitochondria. To cut a long story short, because I don't have the time, I just want to show you that we were able to demonstrate that fumarate hydratase loss really perturbs the mitochondrial reticulum to the extent of generating this hyperfused mitochondria. And this is happening not only at the level of the mitochondrial structure, but also by looking in super resolution microscopy using PK-mito. This also creates dramatic changes in the crystal structure. But more importantly, fumarate give rise to the release of mitochondrial DNA in the form of mitochondria-derived vesicles. And you can see this here. This is, again, a magnification of this swollen mitochondria that we observe in fumarate hydratase deficient cells, but also when we give fumarate to the cells, this mitochondrial DNA is cut off, and we demonstrated that this activates what is called the CTSI cascade. So this is in the summary of how we explain this activation of inflammation in the fumarate hydratase deficient cells. Fumarate accumulation gave rise to the formation of this mitochondrial-derived vesicles that activate CTSI and eventually an interferon response. So I'm posing here what I told you so far is that fumarate hydratase loss gave rise to metabolic adaptations, to oncogenic cascades. But a very important question for us was whether all these signatures were actually sufficient to drive cancer. And quite surprisingly, and maybe in a negative way, although this fumarate hydratase deficient cells have higher ability to migrate, to have this invasion in 3D, when we injected these cells into nude mice, they didn't give rise to any tumor. So this was a blunder. Of course, we expected all these things to at least give rise to transformation, but this was not sufficient. And actually, Lorea Valcarcel, a postdoc in the lab, took the challenge and tried to investigate whether there were other additional oncogenic events that would be required to fumarate hydratase loss to be fully transformed. And so what she did was a genome-wide CRISPR screening. At this point, she didn't look at genes that would kill the fumarate hydratase deficient cells, which are on this side, but actually genes that, when knockout, give the cells an advantage in proliferation. And to cut a long story short, we found among the targets that this protein called HERA, which is a histone 3.3 chaperone, when we knocked it out, actually, would increase proliferation in the cells. Now, I don't have the time to go into details. Again, I just want to give you maybe a conceptual remark. But what we found is that indeed, when we knock out HERA, now the cells injected orthotopically in the kidneys give rise to massive tumors. And this was very important for us because it's the first time that actually we can make cells transformed. And not only give rise to these massive tumors, but this tumor also invade the kidney. So this suggests that we really need HERA loss, at least in these cells, to undergo transformation. How mechanistically this happens? So what Lorea did was actually to perform a transcriptomic analysis, and she found, this is quite intriguing, that the most activated signature upon fumarate hydratase and HERA loss was the activation of MYC target genes, and in parallel, the activation of EMT. And this was also confirmed in another human cohort where we could see the activation of MYC expression. So again, I don't have the time. I hope this schematic will be sufficient. But what we found through additional investigations is that HERA is actually in a histone 3.3 deposition independent manner, is able to exclude MYC from the nucleus. So it's impairing the ability of MYC to activate a variety of transcriptional responses. When we lose HERA, MYC can now activate a transcriptional response, and very surprisingly for us, is not only activating the EMT transcription, but also the synthesis of enzymes that are needed for purine biosynthesis. So in a way, this unleashes this problem probably that the cells have in nucleotide biosynthesis, and the cells can now grow and give rise to fully transformed tumors. So this is the final schematic that I want to show. Hopefully it doesn't be too confusing, but I will try to make sense. I open this. So by looking at what I call here the chronology of fumarate hydratase loss, we were able now, and we are still working on this, to put some of these changes in a time-dependent manner. We know that metabolic adaptations, for instance, in the cells occur very early, but we were able also to understand that there is a very subtle activation of inflammatory response. Why didn't we pick this up in the tumors or in the cells that have been chronically affected by FH loss? Is because the other transcriptional signature are overwhelming. So this is present, but it's overwhelmed when we do gene enrichment signatures by all the others. We needed to go very early, and this allow us to go deeper. And we were able to show that, again, this inflammatory response occurs very early, but is chronic. The other very important observation that we made is that all these responses are actually tissue dependent. We found that there are tissues that are permissive, whereby the loss of fumarate hydratase is tolerated, maybe at least in part, because fumarate is allowed to accumulate to a non-toxic level. But there are other tissues that we call non-permissive, whereby fumarate reaches very high levels, and the cells simply die. And eventually, the last thing that I want to mention here is that we found that all these changes do not seem to be sufficient to drive transformation, but we need other events that are still unclear in the human cohorts. But at least from an experimental point of view, we were able to find the HERA loss and the activation of MYC transcriptional response as a prerequisite for transformation. And with this, I would like to thank all the people that I hope I mentioned during the talk, but there were a lot of people in the lab in Cambridge that I acknowledge, and a few of the lab members that helped all this work, and all the collaborators, in particular Julian Prudent, for all this work on this mitochondria-derived vesicles, and all the others. But more importantly, I thank you for bearing with me for 45 minutes. I hope this wasn't too much, but I am happy to take any questions, and I'm very looking forward to hearing from you.
Questions?
Great talk. I'm curious just about thinking about the first part of your talk about the metabolic liabilities. I'm curious to know whether you guys have looked at other metabolites with primary thiols and whether they might react with fumarate similar to cysteine or glutathione, and whether those might lead to other metabolic liabilities.
Yeah, so you're asking whether, for instance, we gave the cells N-acetylcysteine, which can bind to fumarate, for instance, or
Yeah, or just other primary thiol containing metabolites.
we
Maybe like CoA or anything else like that.
Yeah, good point. So we expected CoA to be somehow reacting to fumarate, but we don't have evidence of this. So when we do the metabolomics, maybe because some of these adducts are not annotated, the only one we could find was the adduct between fumarate and cysteine and glutathione. But yes, it's a very good point. I think it might be good to do a proper and in-depth metabolomic analysis. I would love to do that. That's a good point.
Beautiful talk, Christian. I had a
Thank you.
question about the tissue specificity. So have you looked at whether or not that's also cell intrinsic or cell autonomous? So in small intestinal cells, I guess, whether or not you still have
question.
this. And if so, is it because there's increased sensitivity to, I guess, cell death inducing factors, or you think there's metabolic adaptations that happen in kidney cells that do not happen in the small intestine?
I think so. Personally, I think the second option is the one that I'm encouraged to, to believe. I think in the kidneys, for maybe the intrinsic ability of the kidneys to secrete fumarate, or having all these metabolic pathways that are very good in, tolerating fumarate, they can survive. The small intestine instead is is an absorbing organ, so they take nutrients instead of secreting. So maybe they are not well equipped to this accumulation of metabolites. I mean, this is a completely unsupported hypothesis. It could be, for instance, I mean, I say cell death, but we don't know actually why the cells die. There is a postdoc working on this at the moment. But yes, this is all, and I don't even know, to be fair, whether this permissiveness that we are talking is exclusively metabolic or maybe transcriptional. There are other factors that are, implicated. This is very early time for me.
Just a quick question about MDVs. So, or at least the DNA release. So work from Thomas Langer has shown that if you have imbalanced pyrimidines, you do also end up with DNA release.
Yes.
So just curious whether or not you investigated that in the context of the fumarate hydratase loss.
Yeah, this is when, we talked with Thomas, we thought, okay, that is the explanation of the release of mitochondrial DNA. However, maybe I can tell you what we did to exclude that, is that we treated cells with fumarate, just pure cell-permeable fumarate, and we found that this occurs, the formation of MDVs occurs even in the absence of any nucleotide imbalance. So it's not a direct assessment, but we excluded that indirectly. Thank you.
I just had a follow-up question about the MDVs. What's the fate of them, and how is it that they lack TOM20?
Yes, this was a big surprise. At the moment, we don't know. I think Julian Prudent is trying to figure this out. At the moment, we are not working on this because it's a very complicated topic, and still the field of MDVs is, it's not there yet for the tools. We probably will get there, but it will require some years, I suppose. So to ask about the TOM20, we think these MDVs are without outer mitochondrial member, but as you correctly pointed out, it could be that they are devoid of TOM20, but there might be other members of the outer mitochondrial membrane that are there.
I have a suggestion for that. This is not my question is still coming, but this is a suggestion.
Okay.
TOM20 is one of the first proteins to be degraded when there is uncoupling. This was shown actually by David Chan many years ago. So it could be that those MDVs would have contained TOM20, but you get uncoupling going on there for some reason.
In a localized manner, you mean?
Yeah. Any drop in membrane potential that you see, and there could be a drop in membrane potential if you have impaired TCA cycle.
Okay, so we check the mitochondrial potential in these cells, just globally with TMRE. Actually, the mitochondrial potential is elevated, so there is hyperpolarization in this, but it is due to a combination of changes in complex 1 activity, protomotive force, and also the delta pH. So because
ATP synthesis goes down?
Actually, the ATP synthesis is the same as the wild type, but there is an issue with the delta pH due to the accumulation of fumarate that changes the overall protomotive force. We calculated this a few years ago because I was also surprised to see hyperpolarization, but this is what happens.
That was a really great talk. I want to pick your brain about dimethyl fumarate use to treat multiple sclerosis.
Yes.
And whether your findings that fumarate promotes inflammatory response, how you reconcile that with its use as a treatment.
Yes. So there is a big, thing to mention here in the field of fumarate esters. It's their reactivity, and DMF is much more reactive than fumarate from a chemical point of view. I'm talking about the chemistry here. Due to this dimethyl moieties, fumarate reacts with everything, all the cysteine in the blood, even when the drug is taken. So, and this gives rise to a very powerful antioxidant response. So I think there is evidence out there that the effect of DMF is more mediated by NRF2 than any other functions of fumarate per se. So DMF might be a completely different beast. Based on, also on our experience, if you give DMF to cells, you deplete glutathione in minutes, even at very low dose. But if you give monomethyl fumarate, which has only one of the two carboxyl group esterified, it's already less reactive, and the situation of succination is very different.
Any other?
Nice talk, by the way. So you showed that when you knocked out hemoxygenase 1, right, the cells die.
Yes, correct.
So just thinking about the tissue specificity. So I wonder whether or not the other tissues that do not develop tumor, whether or not they have a different regulation of hemoxygenase 1. That might be the reason why.
Absolutely.
And the question I have is, in the kidney, I do not know which cell type actually develops the tumor, because it looks to me like the renal proximal epithelial cells, because I saw that it's in the periphery. Yeah. But do you know whether it's exactly that?
This is another big question in the field. The cell of origin of this tumor, I don't think is being characterized at all. As you pointed out, it seems to be the tubule, but we don't know. We would like to investigate this more closely. Maybe, yeah, it's a, at least to me, outstanding question. We don't have an answer.
I think we have a question from the Zoom. Any thoughts why glutaminase inhibition did not work as a treatment strategy?
Yes, so based on our experience, when we use glutaminase inhibition, we kill both the knockout and the wild type. So I think glutaminase is probably too much of a general drug, and it doesn't achieve this synthetic lethality as one would expect. This is based on, experiment that we have done. Initially, we thought, okay, glutaminase inhibition will be the best, but it is not.
Yeah, I have a question, but before my question, I want to remind everybody that we have the LA Best event coming next week. So if you want to participate, you have to register online. There won't be registration on site. And the metabolism theme has 3 specific events that will be interesting for you during this, in addition to a lot of really good food there. But this is the opportunity to connect with biotech and pharma in the field. So don't miss on this opportunity for so the registration there is $750 unless you're UCLA, and then UCLA student is $100, and you can ask your PI, so it's free. So don't miss on that opportunity. So okay, to my question. So I know I looked at the slide that you showed from in the intestine, and it looks like there is some ballooning, meaning there is some fat accumulation.
Yeah.
So lipid droplets. So I guess the fat is absorbed, but then it is not released to the blood, right? That's probably. So what's the mechanism for that?
We don't know. So actually, glad you mentioned this. When in Cologne, there is the lab of Manolis Pasparakis and Sander Trifunovic have actually demonstrated when you have a mitochondrial defect, they use SDH knockout and complex COX-10 knockout. You have a similar accumulation of fatty acids. And this is with a mechanism that is not characterized, but it seems to be dependent to from a Golgi derangement that you have. And so you cannot form the chylomicrons. So you get, as you say, absorption of the lipids, but then instead of releasing the chylomicrons as this tissue, the cells will do, there is an impairment in the transfer of the lipid droplets into the Golgi, ER, and this is blocked. And this is happening in those tumors. In those tissues. Whether this is happening in this condition, we don't know. But the postdoc who actually published that work is now working in the lab.
Yeah.
So we're trying to figure that out.
Okay. Great. Thank you.